Thank you for your kind introduction. So, good afternoon, all. Good afternoon. Okay. So, actually, I was supposed to speak in a different session yesterday. So, because of my other commitments, I could not join here. Then, so they included me here. So, but my slides are partly mixed now. So, basically, I'm supposed to talk about more of chemistry and particularly where CSAR contributions for Indian pharma. That's what my supposed to be title. But ultimately, my original research is directly relevant to national product research. One way I am good that so now I could change my some of my later slides. I'll be brief on this particular contributions to Indian pharma, but I'll spend little more time on uh, national product research, what we do in my own research group. Later part of my talk. Right? I don't know how many of you are aware our CSIR, that is a Council of Scientific and Industrial Research. This is one of the largest organizations in our country. So we have 37 laboratories across India, starting from Jammu Kashmir to Kerala. So we have 37 labs. So if you look at the colors, so physical sciences, chemical sciences, biological sciences, information science, engineering sciences, there are different themes we work. They're all part of science, but different domains. So like if you look at all this red color, all chemical sciences, of course we do have within the laboratories we have multiple departments where we work on chemistry, biology, and different thing materials. But that is majorly focused area is on chemical theme, right? So this is one of the biggest organization. Always president of this society is our prime minister, and vice president is our science and technology minister. So we have Dr. Kalai Selvi. She is a DG of Director General of. This is basically she takes care of all the labs spread across India, right? So I started my career at NCL Pune. That's a National Chemical Laboratory Pune. I spent quite a bit of time after my PhD postdoc. Then I moved to northern part of India. That's Indian Institute of Integrative Medicine. That's called RRL Jammu. So later I spent some time in CDRI, that is Central Drug Research Institute. Again, now I moved back to Hyderabad, where presently we are at Hyderabad, IICT, that is Indian Institute of Chemical Technology, right? So we all know, I think by everybody knows Hyderabad is known for pharmaceuticals. We call it as a pharma hub, this particular city. So because most of the drugs exported from like India is of course major drugs are being imported. In that Hyderabad is more known for that. So let me I can give you one example, particularly in USA. Out of 3 drugs prescribed, 2 comes from India. So where Hyderabad contributes more. That's a big contribution. We feel proud about that one. So again, how it came, how we reached this stage. This is a proud moment for us, but how did we reach? So if you look at very early days, 1970s, and most of you are not born, not even there that time, 1970s, we are mostly depending on outside countries, right? Almost non-existent of these pharmaceutical companies in India. So in 1970, this new patent laws we basically we adopted. We started our own patents, issuing patents. That's where 1970 Patent Act. So that's where domestic companies started. But again, there was no expertise. So that's where CSIR labs came into picture. So they contributed a lot in this domain, particularly Hyderabad company, Hyderabad, that is RRL. Presently, it is the Indian Institute of Chemical Technology, CDRI, that is Central Drug Research Institute. So we have National Chemical Laboratory, multiple labs across country. They worked on this process development. We mostly call it as a generic drugs. Like basically after out of patent, they become generic drugs. So that's where all laboratory scientists worked. Then they collaborated with industries. Finally, they've in 1990 and 2010, that is a boom for all pharmaceutical industries. That's where you can see like all the skills we generated, like we established, we became very popular, and also we became self-sufficient, and also we became even exporting also our medicines. From 2010 onwards, so number of patents also increased, and particularly National Laboratory CSAR much more became robust. So now it became a situation that now companies are doing very well, even less dependence on our institutes. So that's where there are contributions from national laboratories. In particularly Hyderabad, there is IDPL used to be there, and this RRL Hyderabad, and also there is Osmania University, Central University. These are the major players they played. Of course, major credit goes to CSR labs, right? So just to give you a couple of examples, if you look at AIDS drug, that is AZT, that's a zidovudine. So originally originated from IICT and in collaboration with basically this is development, process development. So where we reduced cost, it became $1 per pill. So Cipla took, basically collaborated, and they made it available for across the world, $1 for the drug. That is actually, that eradicated most of our AIDS across the globe. So that's a big help we did from CSR labs. Similarly, so Imtech Chandigarh, that's streptokinase. That is again as a clot buster. So that these are all some of the major contributions. If you look at amlodipine coming from NCL Pune, so that is again that a calcium channel blocker. So that again there, I don't know whether how many people know about this resolutions. If you have Resmic mixture, you can make it plus and minus. So they made both S amlodipine or amlodipine. So one of them became more popular. That's so Cipla took that particular contribution from NCL Pune, right? Like that, there are other examples like misoprostol. Again, this is coming from IICT and Avra collaboration. Avra is a A.V. Ramarao company. Earlier used to be the director of this particular institute, Indian Institute of Chemical Technology. So a collaboration, they delivered this misoprostol process development. Similarly, nevirapine and also CDRI, that's ephedrine molecule. These are Malladi drugs. So they took license from CDRI. They produced tons of this compound. So these are all some of the contribution I'm just showing you here. But again, very recent contributions. If you look at, there is a paracetamol. I am sure everybody knows about paracetamol. You need tons of material. That one. That's again, many companies are produced, but this is like cost-effective, continuous flow, eco-friendly process developed by NCL. That's again transferred to different companies. So another compound, azelaic acid. This is particularly used in cosmetics and also treating acne. You see some pimples. So that particular drug also being that material, that azelaic acid was analysis. They use the process. So that is also transferred to different companies, right? So just to give you a very recent example, one of the intermediate called hydrogen hydrate. This is used for as a starting material for many APIs, agrochemicals, and also as part of rocket fuel. We use this particular chemical. So right now, global demand is about 200,000 tons per annum. So India requires almost 20,000 tons per annum. So we always depend on outside the country. So there is a need for India to develop a good process for this. So that's where IICT some time ago started process, bench level, then scale, then pilot scale. Then finally, along with the GSCL, they established a plant here in Gujarat that can produce 10,000 tons per annum. So recently, I was in October 2022, our Prime Minister dedicated that facility to the nation. So very recently, I was part of this first production of first commercial production batch. They shipped out. Flagging ceremony, I was there in this particular place. So these are all some of the contributions we have been making to the country and also to the people, to the world, right? So then another recent example is in COVID time, we all suffered because of lack of medicines, of course, lack of awareness. So many things everybody went through this one. We desperately needed medicines. That's where we came up with this process with the favipiravir. Again, we collaborated with Cipla. We marketed this drug, particularly very early stages of our COVID pandemic situation, right? Another one is Covaxin. So Covaxin came from Bharat Biotech. So I'm sure many of you must have taken this Covaxin. So each vaccines that has multiple ingredients. So one of the component, that adjuvant, that is TLR78 agonist molecule. So that is also as part of very microgram present, but there is no production of that, that was not available. Where IICT made that compound, kilos of material, and we supplied to Bharat Biotech. Then Bharat Biotech included that as part of Covaxin. And so now whoever took Covaxin today, there is some small component, the micrograms of this TLR78 adjuvant is there. So that's where whoever took Covaxin, there is small component of IICT also there. So that's really proud moment for all of us. So that's where all contributions, right? So in addition to that, during the COVID period, I was in IIM Jammu. So we are part of some of these clinical trials where we are along with IICT and one company called Laxai. So we are doing clinical trials for repurposing of old drugs. There are niclosamide, colchicine, there are old drugs. Again, there are, if you look at this colchicine, there is also natural product colchicine. So we did clinical trials. Right now we are in the evaluation. Of course, COVID also now reduced, so little interest also gone down on that one. So that's where we have been contributing, right? This is another major contribution from IICT is one big deal between private pharmaceutical company and also governmentally funded project. That is, this is Sun Pharma. This is NCE for unmet medical needs. We didn't deliberately we did not disclose much details about this one because we are under agreement with the Sun Pharma. This is one of the biggest deal between government organization and private partner. So you look at the numbers. So including milestones, about up to 240 crore deal between company and IICT. That's one of the biggest deal in our country. So that's some of the contributions we have been working, and we have filed several patents even after the deal also, right? Again, currently what we are doing at IICT and CSR labs, IICT has a nodal laboratory because ours is a chemical laboratory. We are nodal, and multiple labs like CDRI, IICB, IIIM, NCL, NIST, Jorhat, NIST, Tirunantapuram, and IMTech, URIDIP, these are all together. So we are working on a mega project, that API mission project we call it. So where we are developing the processes for affordable health. So that's where these are some of the molecules listed here. So most of the labs are working in process development. And right now we are also thinking about this environmental sustainability analysis. That is something which is going to be the mandate for the future. That's where our IICT contribution, one of the group in our institute, led by Dr. Venkat Mohan group. So we are also doing this analysis of all the process development, what we have been working on this. So then, of course, with the help from our sister organization, URIDIP, also as part of this one. So these are some of the molecules we picked up. Of course, we always take some other molecules also, but these are the listed molecules which actively involved in different laboratories, right? So if you look at whole pharmaceutical value chain, starting from drug discovery, drug development, manufacturing, distribution, sales and marketing. So literally IICT CSR labs can contribute every level. If you look at this drug discovery, we have all these very established laboratories are there in biology laboratories, and also chemistry, drug development, and also manufacturing, where process development is a major activity. These are some of the activities what I would like to tell you all this. We can contribute every part of this value chain in the pharmaceutical industry, right? So now I'll move on to very relevant to this particular theme, this particular session, that is a natural product for drug discovery. Again, I don't have to dwell much on this one. Our previous speaker, Professor Jacek, already talked about it, why it is important, natural products. So more than 40% of marketed drugs which are available today in medical shops, some relation with the natural products, whether it's directly they are natural products or some of the derivatives of that natural products. That's the kind of relation what we see derive medicines and natural products. So because why it is so important, why do we so important natural products? One is the scaffold diversity, because structural diversity, they are so different in natural products, right? And typically have a higher molecular mass, larger number of sp3 carbons. So more stereo centers, more specific to the one particular proteins. It's like, some of the heterocyclic aromatic compound, they're all flat structures. So selectivity is less. Whereas if you go for natural products, you see more stereo centers like sp3 centers you call it. So if you have more sp3 center, more specificity, selectivity will be there. That's the advantage with the natural products, right? So one of the very famous example I always show in some of my lectures, that is a Reserpin. That is again, India can take this credit. This is one of the early natural products we isolated from our, this is, what is this called, Raulfia serpentina. This is, Sanskrit name is Sarpagandha. This is one of the natural product isolated. This is so many known people, I mean like well-known people like R.N. Chopra, this Mukherjee, and APC Rai, and Gannath Sen, this Karthik Chandra Bose, this Siddiqui. So these are well-known, established people in this natural products and pharmacology area. So they worked decades on this particular molecule. So this was originally isolated in 1952. Of course, this is being used, it was used for CNS disorders and also high blood pressures. So this, I was also fortunate to work on this synthesis of this molecule during my PhD. So I did PhD in one of the laboratory in University of Hyderabad. Of course, later we moved to ISC Bangalore. That's Professor Govardhan Mehta's lab. That's where we worked on total synthesis of these molecules. I worked on this particular natural product. So when I moved to IIIM Jammu, I was happy to see that farms there, this Raulfia serpentina being grown in our IIIM fields. So that's some connection with my personal research experience, right? So just to give you one more example from IIIM Jammu, this is it's a disoxylum. From disoxylum, originally they isolated rohithikin. So rohithikin, if you make derivatives, finally they ended up with this IIIM 290. That's very potent answer compound. We just got approval from DCGI to conduct phase 1 clinical trials. So that's some of the, this is basically directly natural product from the plant. So rohithikin, natural product, from natural product to derivative, that 290. The difference is, if you look at this one, the dichloro, that aromatic ring, that's basically, that is only the addition with respect to rohithikin. So that's that's something, one contribution from IIIM Jammu, right? So coming back to my personal research group, I started my career after spending few years in industry. I moved to NCL Pune. That's where I started my own personal research. That is a total synthesis and medicinal chemistry. What we do is, so like Professor Jacek, so many people isolate. Of course, right now very few people are there, isolation of natural product in the country and also in the world. Of course, we require more people. It's difficult to, it's one of the challenging area. So like people like me and who are synthetic chemists, what we do is we focus on making those natural products in lab. So that's where we call it as a total synthesis of natural products, right? So we make this molecule, we make libraries of molecules, then we use them for some applications, particularly drug discovery and also crop protection, insect repellent. There are things, some of the things we apply after making these molecules in laboratory, right? So why synthesis? Of course, we talked about why these natural products are important. So, but why I'm looking into some more further sharpening this natural product synthesis. Why it is so important? Because most of the natural product which directly get from the nature, so may not be sufficient quantities. We isolate in small quantities. Some quantities are very much available, but some of them are very, very minor components. But where you make synthesize, you can make large quantities. In addition to that, you can also improve their properties. Some of the compound natural products may not be soluble, they are toxic, they are not appropriate for becoming as a drugs. But if you derivatize, if you make analogs of that natural product, you can optimize. So there is something called, I'm sure many of pharmacy students understand this SAR, right? Structure-activity relationship. So you can do structure-activity relationship with these molecules. So that's the advantage with this synthesis, right? More than that, so personally I believe that the people who work on total synthesis, they get exposed to different reactions. They are well trained in their chemistry. Right? This is one of the example when I was in my industry. I was working in Tata Advinus in collaboration with Novartis. So we identified a molecule. So, but before that, I'll tell you what is the background. This is one of the natural product. If you look at this one, can you can, yeah. So this particular natural product. This is called fluorescein. It showed anti-diabetic effect. That means reduces sugar level. So then structural modification, they finally identified this T1095 molecule. This is later they found that mechanism of action also that works through sodium glucose co-transporter inhibitor. So that's interesting mechanism that later they found it. So but there is, if you look at this sugar moiety and this aromatic ring, so there is a link, O glycoside we call. So in our body we have glycosidase enzymes that can cleave that bond. So then this is also, because of that it will become unstable in a body. So later they came up with a molecule. If you look at this molecule and this molecule, there is that is called directly linked. There is no oxygen between the sugar moiety and aromatic ring. That's called C glycoside. Glycose means you know sugar. So C glycoside. That is, they are stable to glycosidase enzymes. So that's where one of the drug dapagliflozin came into the market, and later other gliflozins, canagliflozin came into the market. So during that time I was at Novartis, I mean like in Tata Advinus, along with Novartis we are working. So that's where we identified a molecule called LIK066. That's a molecule. I was project leader for that. So that's later it is named as a licogliflozin. So that's right now it's completed phase 2 clinical trial. If everything goes right, it can get into the market in few years later. So right now it is also being evaluated for NASH and also this polycystic ovaries patients. So this is some of the satisfaction you get. So you design molecule with the help of a chemist, you make that molecule finally get into the human. That's real satisfaction for any scientist to do that work, right? So in this is a snapshot of molecules what we synthesized in my research group at NCL Pune. Mainly most of the work is done in NCL Pune. If you look at these molecules, so there are different chemical structures, but many of them have a potent biological activity. So what we do in my research group is we make these molecules and we connect with different biology groups. Then we create value for them. I'll show you one or two case studies. So if you look at some of them are really tough, challenging tasks. If you look at this molecule called solamonomide, so we have working for more than a decade. So still we are continuously publishing and a lot of work being generated on that molecule, right? So I'll just show you two examples from my list of natural products. This is one is something called cladosporin. This is a very potent anti-malarial natural product. If you look at this compound, this is originally isolated in 1971, right? So, but recently it showed some interesting activity, that is showing as a lysyl tRNA synthetase inhibition. That's recently they found the target. So that's where we became interested on this molecule. So then because of potency and also very good selectivity when compared to parasite versus human enzyme, more than 100-fold selectivity. That's where we became interested on this molecule. So just for the chemist, if somebody who understands chemistry, there are 3 chiral centers in this. So this is 1, 2, 3 chiral centers. If you take 2 to the power of 3, there are 8 possible compounds are there. That means there are 8 possible compounds one can make. You can make 8 different compounds and make these compounds and put them into biological assays, like enzymatic and parasitic, and also what is the information you are going to get from this? Like structure-active relationship. Just not structure-active relationship. There is something called stereo-structure activity. So we added a name in addition to SAR, SSAR we call it. It is stereo-structure activity relationship. So that's where we added into that one. So next goal was to make this compound. Again, I'm not going to the synthesis. We started from this dimethoxybromobenzene. We went through few steps. Then we made fragment B. Then fragment A and fragment B, both we combined using this. There is something called Grab's metathesis. We made this compound and we did further transformations. Finally, we made Clado 1, Clado 2, Clado 3, Clado 4. 4 compounds we made. Similarly, we used other epoxide. We made another 4 compounds. We crystallized all compounds. We proved without any doubt all the structures. So once we prepared all the library of 8 compounds, next job is to do this biological assay. That again, since I am a chemist, I need to collaborate with biologists. Accordingly, Dr. Amit Sharma from ICGB New Delhi and Dr. Dhanashekaran from NCL Pune, we collaborated. And what we did is we took this library, first initially thermal shift assay, then parasitic growth inhibition, and finally enzymatic assay, and finally active isomer. That's what we did. In addition to that, we also did co-crystallization with this enzyme, lysyl tRNA synthetase, and our compounds, they're all incubated. And we found that 2 compounds are co-crystallized. We call it them as a 12 and 16, that is a class A compounds. And similarly, other compounds, when they were incubated, they could not crystallize. So that's where whichever is co-crystallized, those 2 compounds turned out to be the best compounds from our study. So what we try to show is, stereochemistry is very, very important in drug discovery. It's not that first time we are showing, but using this particular scaffold and using this particular enzyme, so we showed that stereochemistry is very, very important for drug discovery. So once you make this one, now next job is you have to do the structure-activity relationship. For that, you need to make multiple compounds. So we made several compounds, more than around 30 compounds we made. Again, we set up that screening. We found about 8 compounds are better than original one. And also further structure-activity relationship, further SARs led us to that Cl2 compound. Again, always we don't have to look at only potency. So more than that, we have to look at druggable properties. Druggable properties means like how it like solubility, AUC, like all the PK parameters also has to improve that one. So that's for that purpose, what we identified is Cl2. So we put this, if you look at this original compound and present compound, the difference is only addition of this hydroxy group. So that hydroxy has different interactions in co-crystal structure. In addition to that, we also have some of the druggable properties like microsome, liver microsome, like human and mice we measured, and also solubility also improved. Number of, because there are again parameters for druggabilities. In overall, what we did is overall drug likeness we improved from when we moved from cladospore into that lead compound. So that's what we did during our SAR. Again, further, just to summarize that one, that particular project. So now I'll talk about one more project. This is hunanamycin A. It's another natural product. This is again antibacterial compound, which is acts on this Salmonella enterica. So if you look at the structure, there's something related to that riboflavin. So based on the structure similarity, we believe that this goes through riboflavin synthase inhibition. So why it is so important, riboflavin synthase inhibition? Because in riboflavin we get humans and animals they get from food, whereas some of the bacteria they synthesize using this riboflavin synthase enzyme. So if you selectively arrest or growth of that riboflavin synthase, you can selectively kill the bacteria. That's the advantage with this particular target. That's where we became more interested, this particular compound. So if you look at some of the parameters of this molecule, these are again fitting very well with the Lipinski rule 5. So again, pharmacy students very much aware this Lipinski rule of 5, right? So that's whenever we pick up molecule, if it follows Lipinski rule of 5, we pick up those molecule or we give preference to that molecule. This is very well fitting very well. So let me also tell you that this was isolated from marine bacteria. Originally they isolated less than a milligram, 0.8 milligram they isolated originally. So that's not sufficient to do any further biological activity. That's where we wanted to make this compound and study further. So for that, what we did is we took this starting material. That's very easy to make. If you take corresponding diamine and oxalic acid, you heat it, you get that that particular compound. Then you prenyl bromination, then Friedel-Crafts alkylation, that cyclization. Then install this sugar like this allyl derivative, osmium tetroxide media hydroxylation. You get this hunanimycin. So that's where we made more than 80 milligrams. Originally it was isolated 0.8 milligram. Now we can make more than 80 milligrams. So even then, we were not happy with that. So because number of steps are taking more, and also this cost of starting material is 25 grams is a 450 US dollars. If you look at other side, riboflavin cost of 25 grams, 32 US dollar, very cheap. So we thought, why can't we make natural product starting from there? So that's where one of student joined in my group, Rahul Shingare. So what we did is we took the riboflavin, we did some transpiration, like transformation using sodium hydroxide H2O2. So you get this, basically this ring get cleaved. So once you get cleaved, prenylation and cyclization with the Friedel-Crafts alkylation, you get this compound. So 3-step sequence, gram scale synthesis, no column purification. It is like a biomimetic synthesis and 96% purity by HPLC just for washings. So this is one of the beautiful synthesis. Now we can make multiple grams of this compound. Just take riboflavin, and if you do few steps, you will get this unanimycin more than 10 grams. Look at the beauty, like less than 1 milligram from natural sources we isolated, like people isolated, but we could make more than 10 grams of this compound. Next what? So we also wanted to understand this mechanism of action for this molecule. So although we have some hypothesis because of structural similarity, it is going through riboflavin synthase inhibition. So for that proof, what we did is in penultimate step, we incubated decreasing concentration increases the production of riboflavin. So that's indirectly we showed that it is indeed going through riboflavin synthase inhibition. So next job is again to do all similar SARs, that is a structure activity relationship. We made several compounds, more than 70 compounds we prepared, and we finally identified 100548 as one of the lead compound, and this is some of the details are published in one of the journal here and also filed patents. So this is a 8-fold potent than original natural product, and also better PK profile, and also we have LD50 is like safe molecule, is more than 2 grams per kg, and right now basically it is also can be used as a food preservative. We are exploring that idea. So last 1 minute I'll take. So you all know about this cannabinoids, right? So, right, this is there are 2 molecules. One is CBD, THC. There are so many molecules isolated from this cannabis. So this one molecule recently caught my attention, our attention, that is a dimer. This particular half portion is CBD, cannabinoid. This is a dimerization, this is the product. This is what they isolated recently. This is so poor yield, I can tell you, that's about 500 kg of material, cannabis material they use, and they isolated like couple of milligrams of this material. That means you need lot of cannabis material to get to that dimeric material. This is challenging, right? So because first of all, you cannot access to this material if you, particularly in this only if you have license, government license only you can do. That is only possible in few places. In IIIM Jammu it is available, we can do that one. And in some countries they completely ban. Even if you try to do it here, so police will put you in jail. So that's kind of restriction we have. Although we have a lot of medicinal properties of this particular cannabis material, right? So one option is dimerization of this molecule that they tried, they did not work out, and so we came up with some synthetic procedure. What we did is we took volvetol, we did bromination, cyclization, like dimerization, you get that one, and remove that bromine from there, and again what you do is react with this particular terpene moiety, you get to this material. So we could make a dimer compound. So now, like look at this one, from 500 kg of cannabis material, they could isolate couple of milligrams. So now we can make as much as possible this CBDD compounds. Like that we made some of the derivatives, different compounds we made. So right now we synthesize this compound. So we are in the process of doing biological activity of these molecules. Again, these are some of the reactions are not published. And again, what I'm trying to conclude here is we work on application-oriented organic synthesis in my group. Then we know only synthesis, but we connect with the biology groups and we make create value for them, and finally we make molecules which are useful for solving several problems. So hopefully some of the leads what we identified in our group can potentially become useful in the future. So that's kind of what we do. Again, this is my presently research group at Hyderabad in IACT. Again, all these people who are outside the box, they're all graduated. They're all my first set of students. Some of them they took up already their personal their own careers, and some of them are doing postdocs. But right now, currently, my research group in IACT. So we are last few years we got a little disturbed from NCL, from Pune to Jammu, Jammu to Lucknow, Lucknow to Hyderabad. So now we are back to one place. Hopefully, we'll continue to do whatever we have been doing, some of the research here. Again, finally, I would like to thank my organizations and funding agencies and collaborators who really helped us in creating value for the molecule what was synthesized. Thank you all.
Can you please comment on the future of that riboflavin derived molecule which you are positioning as a food preservative? You have invented one riboflavin derived molecule as food preservative. So why that positioning?
Okay. So see, this is this is anti-salmonella compound, right? So anti-salmonella compound, these are all useful, particularly in particularly meat processing industry. There's a lot of problem that infection with the salmonella. So what we are trying to do, since it is very safe molecules, we are trying to work with that industries whether we can use this molecule as a preservative or anti-infective compound. Basically, it can anti-salmonella compound. So that's where we are trying to do that one. So that's right. Okay, we have to see, sir, because right now we are only looking at salmonella, that one.